In this video, we're going to be talking about how inhibitory neurotransmitters are going to be inhibiting action potentials through allowing certain ions to enter the postsynaptic neuron. And there's two different scenarios, or enter or exit the postsynaptic neuron. And there's two different scenarios. One's where a certain type of ion enters the cell, and one's where a certain type of ion leaves the cell. And both are going to be acting to inhibit an action potential from occurring and thus stopping a signal from traveling through the neuron and traveling to the next neuron. It stops the signal so that there's no more signal being passed on through the body. And this happens in it can happen in different things, but a good example is in drugs like pain medication. If we want to stop, if we have a signal going through that's pain, if we have pain traveling through the body, and we want to stop the pain, then we can stop the pain by stopping the signal. Because if the signal stops, let's see, so the signal can it travels down this neuron and then it gets to this neuron, and if it stops at this neuron and it never reaches the brain, then we're not going to feel the pain. So that's just a good example of when this is happening. And so there's two different scenarios in which this can happen. And also, if you haven't watched part one of this video series, then please watch that first, where I talk about excitatory neurotransmitters. That's going to be the topics that I cover in that are pretty helpful to understanding these. But so starting off with this, we're going to start in the same scenario where we have a successful action potential that traveled along the presynaptic neuron. It got to the end of the neuron, and when it gets to the end of the neuron, calcium floods into the cell. When calcium floods into the cell, and calcium floods into the cell again because there are voltage gated calcium. I'm not sure if they're voltage gated, but there are calcium channels. Actually, they might be. That would make sense. But there are calcium channels that are going to open, and when calcium channels open, calcium is high on the outside of the cell and it wants to spread out. So that means it wants to go into the cell. And when it goes into the cell, it's going to trigger these neurotransmitter globules to move in this direction so that they can fuse with the membrane and be released. So same thing that happened here in the excitatory response example. But there are different neurotransmitters here. I'm not sure if you can tell, but I tried to make them different colors. It's kind of hard to see, but we've got different neurotransmitters here that are going to bind to different receptors. That's why I drew the receptors in a different color than the receptors in the excitatory response example. But we've got different receptors and different neurotransmitters. The neurotransmitters are typically specific to specific receptors, and when they bind to the receptors are going to be attached to or they're going to be attached to or like interact with these ligand-gated ion channels. And one of these ligand-gated ion channels we could say is chlorine. Let's use this example first, where we have chlorine. So we've got chlorine channels here, here, and here next to the receptors. And the receptors, just like before, when we have I'm going to turn up the brightness 1 second. All right. I just want to get the colors right. So yeah, I think that's the right color. Okay, so I have the neurotransmitter is going to bind to the receptor, And when it binds to a receptor, it's going to trigger these ligand-gated ion channels to open. So whenever it binds to the receptor, let's leave this one closed just because. And then let's open this one. And then let's say that that's a neurotransmitter bound to this one as well. And let's go ahead and open that one as well. So again, when a neurotransmitter binds to the receptor, it triggers a conformational change to the this transmembrane protein to allow the ions to pass through. It's going to open it and allow ions to pass through. The ions that are allowed to pass through when it's opened is specific. So there's a difference neurotransmitter here, and it's going to bind to a specific receptor. This receptor, we are going to say, is interacts with a chlorine ligand-gated ion channel to where it only allows chlorine to enter or leave the cell. If we recall, chlorine is high in concentration on the outside of cells. So there are a lot of chlorines outside of the cell hanging out. There's a ton of them. But there's not very many chlorines inside the cell. What does that mean? That means that chlorine, when given the opportunity, is going to enter the cell. So chlorine, I'm going to draw, let's see, I'll just draw one, and then from now on I'm going to represent it as a negative charge because it's one of the main negatively charged particles. So chlorine is negatively charged, and it is going to, when it has the opportunity, it is going to enter the cell. When it enters the cell, there's a lot of repeats from what we were talking about before. It's going to be very similar to what happened with sodium. We're going to get a lot of chlorines here in the dendrite, in the dendrite, and there's lots of chlorines here. And what's going to happen when we get a high concentration of chlorines in here? Well, they're not just going to stay here with some sort of imaginary barrier. Things like to spread out when given the opportunity. So these chlorines are going to start spreading out throughout the cell, and keep in mind the charge. So in the previous example in the last video, we talked about how sodium, when it enters the cell, it's going to make the cell more positive. And we looked at our graph and we said sodium, when it enters the cell, it makes the cell more positive, like that. But when chlorine enters the cell, chlorine is negatively charged. And since it's negatively charged, it's going to make the cell more negative. So it's going to move, it makes the voltage more negative. So it was at negative 70, it's going to be maybe negative 80, maybe negative 90. And so chlorine makes the cell more negative. And let's look at this value right here, the threshold. Let's look at that, where that's located. That's at negative 55, where the resting membrane potential is at negative 70. So if we're at negative 70 and our goal is to get to negative 55 in order for an action potential to occur, notice that when the cell becomes more negative, we are getting further from threshold. We are getting further from threshold. And when we get further from threshold, that means that it's less likely to get to threshold. And if it's less likely, and the only way that an action potential can occur is if we get to threshold. So notice that chlorine is making it harder to get to threshold. And that's really, really important to remember. That when the cell becomes more negative, it is less likely for an action potential to occur because we are further from threshold now. So chlorine, when it enters the cell, it makes it harder for an action potential to occur. When chlorine's entering the cell through these ligand-gated ion channels from a different type of neurotransmitter binding to the receptor. These are inhibitory neurotransmitters, and when they bind, they allow chlorine to enter the cell. And when chlorine enters the cell, we get it starts spreading out, makes the cell more negative, which makes it further from threshold and makes it harder to get to threshold, and therefore harder for an action potential to occur. So these chlorines, they keep spreading out, and then they're going to get to here. They're going to get to the axon hillock region. When they get to the axon hillock, that's really what this graph is showing. This is the action potential or potential of an action potential at the axon hillock. And we just showed that we are now further from threshold. And now that we are further from threshold, it means we didn't get to threshold. We ended right here. We are not at threshold. We have to be up here to be at threshold. So since we did not get to threshold, an action potential is not going to occur. And therefore, one of the things that happens at threshold is the sodium voltage gated sodium channels would have opened if we got to threshold. But since we didn't get to threshold, the voltage gated sodium channels remain closed. So let's zoom in. Notice how in the previous example, with the when we got to threshold, these voltage gated sodium channels represented in orange, they opened and allowed sodium to enter the cell. But we did not get to threshold, so they are going to remain closed. Look how I drew them, that they're closed. So no sodium is going to enter the cell. And since no sodium entered the cell, that means we didn't get here. Remember, we just kind of just stayed here, and then eventually we'll get back down. We'll get back to resting membrane potential. But the voltage gated sodium channels did not open. And since the voltage gated sodium channels did not open, we never got to here. And since we never got to here, the voltage gated potassium channels never have a need to open. The voltage gated potassium channels, so voltage gated sodium channels, we have to get to negative 55 millivolts in order for them to open. Notice that we did not get to negative 55 millivolts. We got even further from it. So voltage gated sodium channels didn't open. And since the voltage gated sodium channels didn't open, we didn't get this big spike in voltage. And since we didn't get this big spike in voltage, we never got to positive 40 millivolts. And since we didn't get to positive 40 millivolts, the voltage gated potassium channels are never going to open, because that's the voltage required for them to open. So notice how because chlorine entered the cell and made the cell more negative, everything else just stops, and the signal basically kind of stops here. I mean, you could say that the signal stops at the end of this presynaptic neuron. You could say that, or you could say that the signal stops here. Whatever you want to do. The important thing is that it doesn't matter how far the signal goes inside of this neuron. What matters is that it never gets to the brain, or it never gets to a muscle. It never gets to whatever this neuron is connecting to. It never gets to the next thing. So the signal stops, and we're not going to get an action potential. So there's no action potential. So the action potential that would have traveled along here doesn't exist, and there's no propagation of the signal anymore. So that's how chlorine can cause us to get further from threshold and make it less likely for an action potential to occur. Next, we have potassium can also work to inhibit an action potential. And let's see how that can happen. Because you might say, wait, well, potassium is positively charged. Why is it not going to help an action potential from occurring? Well, let's think about it. So potassium It is positively charged. You're right. But let's look back at what's the concentrations of these different ions in the cell and where they're high in concentration. We said that sodium, chlorine, and calcium are all high on the outside of the cell. But potassium is high on the inside of the cell. So let's look at how that's going to play a role here in when these potassium channels open. So now at this third graphic, we're going to show what happens when ligand-gated potassium channels open from specific neurotransmitters, causing them to open. So same thing here in this presynaptic neuron. We had an action potential traveled to the end of the cell, and that triggers calcium channels to open, and calcium is going to flood from high concentration to low concentration, meaning it's going to enter the cell. When calcium enters the cell, the presence of calcium is going to cause these neurotransmitter globules to move in this direction and fuse with the cell membrane. And when they do so, they're going to release their contents, which is neurotransmitters, into the synapse. Remember, this region right here is called the synapse. These neurotransmitters are bouncing around, and then eventually they're going to bind to the receptors. So we're going to have neurotransmitters bind to the receptors here. And attached to these receptors, just like before, just like in all these other examples, attached to the receptors are these potassium channels that are going to open whenever a ligand binds to the receptor. Again, a ligand is just something that binds to something else. So when the neurotransmitter binds to the receptor, it opens these ligand-gated ion channels. So they are closed, but then when a neurotransmitter binds to the receptor, they open. And these ligand-gated ion channels, they are specific to specific ions, just like in these other examples. And this one that I drew in this blue color is going to be for potassium. This is going to be a ligand-gated potassium channel. And again, it interacts with the receptor, and it only opens when the receptor is bound to a neurotransmitter. So when this ligand-gated potassium channel opens, we need to think about where potassium is located. We're representing potassium in purple again. So potassium is in purple, and I'm going to draw it as these purple pluses. So if we recall, potassium is naturally higher in concentration on the inside of the cell than outside of the cell. So when given the opportunity, it wants to spread out and it wants to leave the cell. So as soon as these ligand-gated potassium channels open, potassium is going to seize the opportunity and leave the cell. So we have a whole bunch of potassium ions that are all here, and we've got so many of them because there's a high concentration of potassium inside of the cell. And potassium, again, when given the opportunity, it's going to leave the cell. Because it wants to travel from high concentration to low concentration. And when we lose a lot of potassiums here, when a lot of these get not deleted, but when they move out of the cell, and all of these are moving out of the cell, they all moved out of the cell. Well, are we going to just stay with, are we just going to leave this region of the cell with no potassiums? or are the potassiums that are located over here, are they going to spread out? So again, are we going to pretend like there's an imaginary barrier here and all the potassiums that were located over here, is it just going to be a potassium-free zone, or are the potassiums going to spread out? The answer is these potassiums are going to spread out. And so potassiums that were located throughout the rest of the neuron, even ones that are located over here in the axon hillock region, they're going to spread out. And all these potassiums are spreading out to where we have an even concentration of potassium throughout the cell. And most importantly, when they are spreading out, and when they get over here, they're leaving. And when a positive charge leaves the cell, let's think about what would happen. So our cell has a resting membrane potential of negative 70 millivolts. But keep in mind, even though it has a negative overall charge, there are some positive charges inside the cell. But if we remove, and we talked about in this example with chlorine, if we add more negative charges inside the cell is going to become more negative. But the other way that the cell could become more negative is, what if we have positive charges that leave the cell? Now the cell is becoming less positive, which is also the same way as saying it's becoming more negative. So we are losing positive charges, and since we are losing positive charges, the cell is becoming less positive, or in other words, more negative. So from potassium, so the potassium is leaving, and all of these potassiums that were located in this region of the cell, they left. And when they left, these other potassiums in this region of the cell spread out, and they moved to the left to spread out. And when they spread out, they caused the cell, the overall charge of the cell, to decrease, because we lost positive charges. Positive charges left the cell, and now the cell is less positive, or in other words, more negative. And most specifically, we're looking at this region right here, and we said that some of the potassium ions, we had more potassium ions over here, but they spread out to other parts of the cell, and some of them left. And when that happened, the cell became more negative. So let's look at our chart here, and we have, again, our resting membrane potential is negative 70. But when we lost positive charges, when positive charges left the cell, the voltage decreased. It became more negative. All right, and then let's pay attention to to this threshold value. Did we become closer to threshold or further away from threshold? Well, threshold is at negative 55, and we were at negative 70. The loss of potassium, the loss of these positively charged ions, caused us to become more negative, not less negative. So if we just look at where we're at, we are now further from threshold than we were when we started. So potassium leaving the cell through ligand-gated potassium channels causes us to be less likely to get to threshold, and therefore less likely for an action potential to occur. So the action potential is not going to occur if we don't get to threshold. And again, potassium is making it even harder to get to threshold. So since we don't get to threshold, that means that since we don't get to threshold, that means the voltage-gated sodium channels are not going to open. And then since they don't open, we're never going to get to this positive 40 value. And since we never get there, voltage-gated potassium channels are not going to open either. So if we look on our drawing, we have those voltage-gated sodium channels drawn in orange, and we have the voltage-gated potassium channels here drawn in purple. And they're not going to open, they're just going to remain closed because we never got to threshold. All right, the last thing that I want to cover is that if we remember the anatomy of a neuron, let's draw that very quickly right here. If we remember the anatomy of a neuron, we have the soma with the nucleus, and then we have dendrites, and they're going to connect to other neurons. The ones that they're going to attach to the axon terminals of other neurons. So there's other neurons here that are going to attach to them. And there's other ones here that are going to attach to them. Other ones here that are going to attach to that one, etc. All right, so the dendrite, there's lots of, what I'm trying to get to are that there are lots of these dendrites. There's not just one. I only have one drawn, but there's other dendrites here that are going to attach to other axon terminals of other presynaptic neurons. There's other ones here that are going to attach to other presynaptic neurons. And the fact that multiple of them are connected at once means that sometimes we might get an excitatory response and an inhibitory response at the same time. So we could have where I'll draw one in red. So this one here could be an excitatory response, and it causes sodium to enter the cell, where we're going to get more positive charges here. And sorry, I didn't draw the rest of the neuron, but we have the dendrites and then the soma, and then we have the axon here, and then we have the axon terminal, and it might connect to other dendrites of other cells. All right. But again, we might have an excitatory response from some of the dendrites. We could even draw multiple ones. So we could have multiple excitatory responses. Maybe these are all excitatory responses. But we could have an inhibitory response at the same time. And so what might be happening is we might have, chlorine might be entering here, and then we get some negative charges inside there at the same time as we're getting some of these positive charges. So what's happening is the sodium channels opening, having sodium flow into the cell, is going to make us more likely to get to threshold. But the chlorine entering the cell is going to make it less likely to get to threshold. Because remember, the chlorine is making us less, it's making the cell more negative and making it less likely to get to threshold. So if we have a lot of sodium entering in and not very much chlorine, we might still get to threshold because we have the amount of sodium that was entering outweighed the chlorine. But keep in mind, I just wanted to point out that there can be some level of interaction to where we have two different signals for the same neuron, and it just kind of ends up being this averaged out amount to where maybe if the sodium alone was entering, we would get all the way up here. But since we're averaging it out and we have chlorine also, the chlorine, if it was by itself, it would have maybe been like here. And we're going to average the two out to where we're going to get to here. And since we got above negative 55, the voltage-gated sodium channels opened and we had an action potential. But if we have, and this is talking also if those signals happen at the exact same time. But we could also have an excitatory response where sodium enters Sodium enters, we have an action potential. But then immediately after, chlorine enters. And chlorine enters the cell, and then we have an inhibitory response. And so maybe we tried to have another action potential right after, but the action potential that was supposed to occur right after was inhibited. So just keep in mind that there can be some level of interaction. Whenever chlorine enters the cell, it's less likely for an action potential to occur. Whenever potassium leaves the cell, it's less likely for an action potential to occur. And whenever sodium enters the cell, it's more likely that an action potential occurs. And there's going to be some level of interaction between all of these, and the amount of each one that enters or leaves the cell is going to play a major role, but also the time that it happens. Because if they happen at the same time, then there's definitely a huge interaction. But if there's enough time between, then maybe some of the chlorine that entered gets pumped out, or maybe some of the potassium that leaves gets pumped back in, and it's back to baseline. But just keep in mind that there's some level of interaction there, and that in this anatomy of the neuron, just keep in mind that there are multiple axon terminals that are presynaptic cells that are connecting to this postsynaptic neuron here through the millions probably of dendrites that they have. So hopefully that was helpful. I'm going to be posting these notes as well, and you're welcome to look at those. But all good stuff, and hopefully that was helpful in understanding how inhibitory responses play a role in the inhibition of action potentials.